What if people knew when they were going to die? What if people knew exactly the probability that they were going to get Alzheimer's or Parkinson's or ALS? How do proteins encode the future? What if we could use recent artificial intelligence technologies like AlphaFold to not only understand how a protein folds, but how it will change with time to cause disease, and how we might be able to design proteins to stop that process? I grew up on a ranch in West Texas. It was very isolated. I wasn't able to do all the things that my friends were doing. I entertained myself outdoors. I really fell in love with the diversity of biology around me, the diversity of the natural world. So I had a very extensive rock collection, and I loved thinking about crystals and how matter can self-organize in a way that's beautiful and yet will always do exactly the same way every time. And I had no idea that what I was doing when I was, 10 years old, I would still be doing in a different form right now. Being on the fringe is good in a way. I thrive on it. I was working in plant genetics in a lab where most of the people were out in the field all day crossing cotton plants. And I felt a little bit isolated. I was the only one in the lab with a pipette testing whether we can create a new species of cotton by doubling the genome. I ended up going to grad school at MIT, and then again, I had an opportunity to do something that was a bit fringe. I became exposed to the idea that there are prion proteins that are actually good. This was at a time when prions were notorious, like mad cow disease. They were causing epidemics. Now suddenly to think that, oh, this isn't necessarily just an accident of biology, this might be a fundamental piece of biology. It became clear to me that we needed to understand the structures of proteins at a time in the protein's life where we had no ability to see structure. So I ended up coming to Stowers to develop a way to do that. I had to convince the scientists here to come along with me on that journey. I study how and why proteins assemble. Everybody loves solving puzzles, and here was a puzzle where a one-dimensional string of amino acids, a sequence, somehow knew how to fold onto itself into a three-dimensional puzzle. And every sequence forms a different structure. And so this was the ultimate puzzle to me because all of life depends on it. And it was not just a three-dimensional problem, but there was a fourth dimension. Proteins change with time, and that is somehow determined by that same one-dimensional sequence.
This was extremely surprising and extremely exciting.
One of the themes that's emerging from the work in my lab is understanding that there's a trade-off. It seems that we have to have proteins aggregate in order to have immunity to survive viral and bacterial infection. But at the same time, there's a cost. The ability of these proteins to aggregate also causes cells in our bodies to change over time. When we look in the world around us, we see organisms that have somehow escaped that trade-off. There are organisms that have immunity but don't age. So one of the things we're doing is looking at the proteins in those organisms and trying to understand how they overcome that challenge. So there's a purity in nature, and it seems like all the answers are there for the finding. I don't think that people allow themselves to ask, what if we don't have to age? Or what if we don't have to get sick when we get old? Or what if we don't have to get cancer? We wouldn't be doing these things if we didn't have some belief that we're going to answer those questions. What if we understand how a protein folds and changes with time? Then we'd be able to understand how we can stop that process.